She also brought to us an enormous amount of experience working in the East Coast and the Midwest, having done her undergraduate degree in the States, and also worked at the Smithsonian, where she did work with insects, so she brought a lot of information about insects when she came. So we're really lucky to have the kinds of people that, like NIDHI, that come and bless us with a little bit of extra information from around the world. And I hate to let anyone ever graduate, but I know she's going to go on to do more wonderful things, and I'm really excited to hear her story about her butterflies. So, and the hate.
Thank you.
So, thank you.
Thanks for the introduction. Thank you everyone for coming to my talk titled The Effects of Nitrogen and Grazing on Early Season Nectar Resources for the Adult Big Checkerspot Butterfly. I'd like to start by thanking my committee, Dr. Rachel O'Malley, for helping me in each step of this project, and Dr. Trulio for giving me her valuable time in a very busy schedule. Thank you. And my third committee member, Dr. Jay Pasari, for giving me very valuable feedback on my thesis, and Dr. Dan Hernandez for allowing me to utilize their experimental setup. I'd also like to thank David Thorpe from OSA and BTA for allowing me access to their land. My research assistants, Eric Casedo, Michael Hobbs, and Rebecca. And also like to thank all my professors and Dr. Mayat for helping me with the plant identification, my wonderful cohort and my lab group, and lastly, my friends and family. And now plan of how my presentation is laid out today. I'm going to start by talking about the introduction and background, go into the objectives and questions, then into the methods, results, discussions, and then further research and recommendations. Biodiversity hotspots are defined as a region of significant flora and fauna and is threatened by destruction. These regions specifically refer to 35 regions in the world, which have lost about 70% of their original habitat. As you can see, Northern California is one of these biodiverse hotspots. So what is causing this degradation? Current trends in global biodiversity hotspots, rare species, and protected areas show significant biodiversity degradation caused by current and upcoming urbanization. At current population growth rates, there will be nearly 2 billion new urban residents by 2030. This map shows the region of global urban population increase. The darker and bigger the circles get, the higher the number of population increase. Urbanization leads to the conversion of natural habitats that affects the distribution and abundance of species. Urban development causes extinction rates and leads to the removal of a large majority of native species. Most protected biodiversity hotspots likely to be impacted by new urban growth are in countries of low income, potentially limiting capacity to adapt to these anthropogenic stresses. Another challenge of urban growth is that it replaces native species with non-native species. Pictured here is the water hyacinth, famous for clogging about 80% of the Lake Victoria. Urban gradient studies have shown that for many taxa, the number of non-native species increases towards centers of urbanization, while the number of native species decreases. As this global change occurs, disturbances such as invasive species become more widespread. Invasive species are introduced species where they do not naturally occur. They are the second prominent cause of extinction or endangerment. Ecological theory suggests that the amount of resources available in an ecosystem should affect its response to ecological disturbances, like invasive plants. This is an example of Lactuca, an invasive species in Coyote Ridge. Invasive species have shown to benefit from changes from fluctuating factors that influence plant growth. Some nutrients important to plant growth are nitrogen, potassium, phosphorus, and calcium. Usually nitrogen and other minerals are limiting factors for certain ecosystems, but anthropogenic nitrogen contribution has increased the changes of nitrogen in the biosphere, causing shifts in plant species composition, decrease in species diversity, and also altered certain ecosystems. In some cases, providing a competitive edge for invasives. allowing them to outcrowd native vegetation. For example, a study done by Brooks in the Mojave Desert, a low productive ecosystem, showed that increase in nitrogen, in soil nitrogen, increased invasive species, and decreased native annual plant density cover, even though non-native plants comprise of a relatively small proportion of the desert floor. Plant biomass and rates of growth are influenced by habitat productivity. The most direct effect of disturbance on vegetation is to increase available space and light through competition. The effects of disturbances lead to competition, and one such ecosystem is the serpentine ecosystem. Serpentinite rock is the primary source of serpentine soil. Serpentine soils are generally deficient in plant essential nutrients such as nitrogen, phosphorus, potassium, and sulfur, but have higher levels of heavy metals such as nickel, cobalt, and chromium. Serpentine soils are often found in open, steep landscapes with substrates that are generally shallow and rocky, with a reduced capacity for moisture retention. Due to the pressure generated by such edaphic conditions, serpentine soils promote endemic speciation, contributing to unique biotas worldwide with patchy distributions. Many serpentine plants actually perform better in these harsh conditions. They are specialized, hence serpentine adapted plants are not found in non-serpentine soils. Serpentine grasslands have become a model system to investigate the effects of nutrient deposition on invasive species management. These ecosystems have been used as a model low nutrient ecosystem for studying basic ecological questions because of their species diversity and abrupt resource gradients. Habitats with serpentine soils are undergoing drastic changes due to expanding development, mining for heavy metals, and invasive species. Such changes can have drastic impacts on serpentine flora and fauna. Habitats with serpentine soils provide opportunities for conservation and restoration-oriented research to find better ways to manage these biodiverse hotspots. One such ecosystem is the Coyote Ridge, found in the California Floristic Province Biodiversity Region. Nutrient experiments in these grasslands have all shown that exotic grasses respond more favorably to nitrogen additions than do natives. The increase in development and associated traffic emissions has increased the amount of nitrogen in the atmosphere, which has changed the nutrient profile of serpentine soils and created an imbalance of the delicate system of Coyote Ridge. This graph shows the increase in nitrogen from 1991 to 2001. Although it is below the national standard, there has been a steady increase in nitrogen. A laboratory experiment by Volano studied the performance and competitive dynamics of 5 native species of Coyote Ridge: Plantago erecta, Leia, Lasthenia californica, Vulpia, and Cryptantha, versus the non-native grass Lolium multiflora. Species were subjected to different combinations of gaseous nitrogen and solid ammonium nitrate. In increased nitrogen deposition and solid ammonium nitrate, the non-native grass Lolium multiflorum increased in shoot biomass, as did Leia and Cryptantha. Competitive ability measured by relative yield potential showed an increase in RYP in Lolium multiflorum versus all other native plants. These results concluded that non-native species have a competitive ability to outcrowd native species. Comparisons in disturbances in serpentine and non-serpentine ecosystems have helped improve understanding of plant ecological strategies and establish the influence of grazing in many serpentine ecosystems. Grazing has become a major factor in shaping patterns of native and non-native species in many grasslands, yet this management strategy has had variable effects. Fisher studied the effects of livestock grazing in several serpentine and non-serpentine grasslands in California. He measured composition at about 85 grazed and ungrazed sites for 2 years in serpentine soils and 2 years in non-serpentine soils. Grazing did increase total species richness on both soils. However, grazing enhanced total invasive species richness on more serpentine soils and enhance native species richness on serpentine soils. Grazing increased native species richness on, these results suggest that the use of grazing to manage native species diversity in wildlands must be done while considering ecological heterogeneity. Vasari et al. Studied the effects of fertilization and grazing on native species richness in Coyote Ridge. There was no difference between native richness in grazed or ungrazed sites. They simulated realistic nitrogen deposition with low-level fertilization and manipulated grazing with fencing. This was a 4-year experiment to assess the effects of grazing and nitrogen deposition on several measures of native and non-native species dynamics. Results suggested that native species diversity may increase slightly under low-level nitrogen deposition with moderate grazing in Coyote Ridge. However, grazing may not be effective at limiting invasive species cover as nitrogen accumulates in the future. These studies highlight the variation and contrasting effects of grazing as grassland management. California grasslands support a high rate of rare species richness. One such species found in a biodiversity hotspot in Northern California and is declining in populations due to disturbances caused by urbanization is the bay checkerspot butterfly. It is identified as federally threatened. In order to understand bay checkerspot butterfly fitness, studying the nectar resource is important as it could affect fecundity and eventually fitness. The scope of the study is to being able to understand the application of grassland management, nitrogen deposition, and increase and threatened insect conservation. Why study butterflies? Butterflies have become an important model system for expanding basic knowledge in conservation ecology. They have become key organisms for monitoring biodiversity and are one of the first groups in which distribution shifts have been studied due to anthropogenic disturbances. Furthermore, the bay checkerspot butterfly is an umbrella species. Umbrella species are selected for making conservation-related decisions, typically because conserving the umbrella species indirectly protects other species in the ecosystem. For example, Martin Cannon found that the white-backed woodpeckers worked well as an umbrella for a conservation of a beetle species, which in turn helped conserve the deciduous forest in Sweden. Lawner and Murphy suggested that if all sites occupied by the bay checkerspot butterfly were preserved, then about 98% of the native spring flowering plant species would also receive protection. The bay checkerspot butterfly belongs to the family Nymphalidae and is the subspecies Euphydryas editha bayensis. It is a phytophagous insect which feeds on plants. It is also a univoltine species, meaning it has one brood a year. Therefore, it tries to sync its life with its host plants. It has a short time to feed, mate, and lay eggs. The bay checkerspot butterfly is most active in early spring as an adult, the stage where it feeds on nectar resources, which could perhaps dictate the rest of its life cycle. The bay checkerspot butterfly female lays egg masses of 20 to 200 on the base of its host plant, Plantago erecta. Eggs hatch in about 2 weeks. Larvae then feed for 12 to 15 days until they reach 4th instar larvae, then have to diapause for 5 to 6 months. They end diapause, continue feeding, and finally pupate. The fitness model by Kingsolver and Healy shows mating success, fecundity, and survival as the 3 main functions of fitness for lepidopterans. Larval Lepidoptera allocate incoming resources to grow and influence body size. Abundant literature has addressed the importance of larval state of the bay checkerspot butterfly fitness. Larval studies have been key in understanding the bay checkerspot butterfly ecology and conservation. Weiss studied the effects of nitrogen deposition on grazing on the bay checkerspot butterfly larvae. He collected densities of post-diapause larvae during 96 to 98 from 6 different serpentine outcrop locations in the Bay Area. The population estimates in the Silver Creek area showed pattern of initial growth, steady decline, and finally extinction. Overall, all sites had a decline in larval counts as a result of removal of grazing. which enabled the invasion of invasive grasses. Weiss studied nitrogen deposition from various sources depositing on Coyote Ridge. He studied the effects of an increase in nitrogen on serpentine grassland, particularly the larval bay checkerspot butterfly host plant, Plantago erecta. Weiss found that the invasive grass species utilize the extra nitrogen on the serpentine grassland. They utilize it and to outcompete the native species diversity. Invasive grasses such as Lolium multiflorum, Avena, and Bromus utilize this nitrogen and outcrop native species such as Plantago erecta, Miola maritima, Cryptantha placida, amongst others. He studied grazing as a tool for removing some nitrogen biomass. He carried out studies beginning in 1996 and compared grazed and ungrazed sites for 3 years. Plots also showed the differences in plant composition between grazed and ungrazed grasslands. The grazing regimes in winter and summer showed higher densities of the native Plantago erecta and lower densities of the introduced grasses such as Lolium multiflorum. Weiss also studied larval movement in association with their thermoregulatory behavior. He showed that post-dipause Euphydryas larvae are behavioral thermoregulators. They raise their body temperature and maximize growth rate by basking in sunlight, and fastest growth occurs between 30 to 35 degrees Celsius. Larval temperature is greatly reduced by shading and low ambient temperature. Low insolation levels can restrict larval growth, and even when direct sunlight is available. The distribution of nectar plants can influence adult movement patterns and the spatial distribution of egg clusters. Working with Euphydryas chalcedona in Jasper Ridge, Murphy showed that females became more concentrated near nectar sources as the season progressed. As a result, egg clusters are more common on host plants directly adjacent to nectar. While larval food consumption impacts size, the adult Lepidoptera allocate incoming resources to reproduce and grow. Resource allocation varies under environmental conditions as adult nectar sources vary per season. Although several factors determine the effects of resource stress on fitness and foraging, adult nectar source is essential. Sugar intake could potentially increase mating success, fecundity, and survival, as again shown by the King-Solvay-Hayne fitness model. Fecundity in insects is the actual reproductive rate of an organism, which is measured by the number of eggs produced. Actual or realized fecundity may be dictated by the adult nutrition. Adult butterfly feeding has shown to increase net energy and increase fecundity. Murphy et al. Studied the role of the adult bay checkerspot butterfly feeding on egg mass rates. Post-dipause larvae were collected from 2 sites that used to sustain the bay checkerspot butterfly populations, Woodside and Edgewood. All newly emerged adults were kept in the same conditions, but one group was fed water and the second was fed sugar concentrated solution. Females fed nectar produced more eggs than females fed no nectar. The results showed that nectar plays an important role in increasing production of egg masses, which could be crucial for population continuance. Studies have also found a positive correlation between fecundity and adult butterfly nutrition. Larger lepidopterans with higher quality nectar sources generally lay more eggs than smaller females, as shown by an experiment done by Hill. 2 groups of the same adult butterfly species were fed 2 different solutions of sucrose, 50% and 70%. The number of mature eggs produced by the group that consumed 70% sucrose was higher as compared to the group that consumed lower nectar concentration. While nectar concentration is important, timing of adult nutrition availability is also important. Early season flowering resources were important to my study. Shown by Hunsford et al., who studied the availability of nectar sources of painted lady Vanessa Cardley butterfly. The adult butterflies were fed sucrose from the same flower, Lantana camara, fed at day 2 of emergence, and the second group was fed on day 8 of emergence. The results showed that both females had increased fecundity rates as opposed to the group that had no cleaning up on emergence. The group that ingested nectar on day 2 had a 22% higher count of eggs, plus a higher fecundity rate. Cushman et al. Studied female reproductive success by studying